I'm so excited to be allowed to speak today because from morning we've been talking about biodiversity that we see with our naked eyes. But we want to talk about biodiversity that we cannot see with our naked eyes. And we want to also, because if you're talking about biodiversity, it has to be the holistic, the word macro and the micro. And this paper is talking about the microbial diversity of a remote aviation fuel contaminated sediments of a Lentic ecosystem in Ibeno, local government council. Sometime in 2001, there was a spill, just like she had just shown. Of course, we need to mention to you that down where we come from, spills occur almost every day. In fact, there's data that I have that sometime in 2000, we had about 800 and something cases of oil spills, in which case it was by far much more than 2 spills per day. That has not been published yet, but I think maybe by September, that the spill statistics will be published. And so if we have spills occurring almost on a daily basis, then you can be very sure that it's a big problem. And so this paper would present the diversity in a remote 40 years after a spill. What happens to the diversity in the place? I'll take introduction, methodology, description of the study area, look at the culture-dependent, because we had to use both the culture-dependent methodology and then the 16S metagenomic approach so that we look at the holistic diversity in the area. I will look at the results and discussions, and then I'll conclude. Just like we have said over and over again, without overflogging the issue, I'd like to mention that the Nigerian economy relies heavily on crude oil. Our state, like someone had earlier on presented, is the highest state, the state that produces the highest crude oil in Nigeria from the Niger Delta environment. And oil discharged into the environment from various sources she had presented without overflogging the issue. Let me jump. So we're looking at a spill location where we had this. This man was the paramount ruler of Ibeno, late. And so this one took over from him, and this was the chair lady of the local government, as at the time this study was conducted, about 12 years after the aviation fuel spill. When we wanted to start this, and we said, we are coming to study your area over this, and they said, look, everywhere, almost every inch of our community is an oil spill site, because almost everywhere has been affected by crude oil. And so she had shown some scenes of oil spills. Again, the mangrove within the environment polluted, the shorelines, the open waters. You could see oil sheen almost every time, almost every day. And so spill statistics, like I said, is there, but even though not fully published. But on the 8th of August 2001, like I said, ExxonMobil aviation fuel pipeline ruptured, and quite some aviation fuel was released to the environment. And coincidentally, this place is a stagnant water, a lentic system. The water does not flow out, does not flow, it's just there. And so, usually the oil companies will apply to a regulatory body to apply dispersants so that they can disperse the oil. And then when they apply dispersants, the dispersants would, degrade the oil and then, sink it down to the sediment. And so dispersant was used in this place, and nothing else was done as far as cleanup was concerned. This was 2001. So, 40 years after, each time we enter this place and you match the place to take sample, you see oil, fresh oil seen on the surface of the water 40 years after the spill. And you begin to wonder, there's something that we were taught in science, that there's a self-recovery of an environment when there's a pollution over time, even if nothing is done about it. But 40 years after the spill, 12 years after the spill, when I studied this, I realized that there's still residual oil in the environment. So as a microbiologist, I was bothered. What about the indigenous microorganisms in that environment? What are there? Can't they degrade the crude oil? And over the years, can this place recover? You know, the issue of ecological resilience. And so we had to design, this is the site. You could see, anytime you enter the place, you could see oil shade, just match on the place, and then you see. And so, at almost every location, almost different locations in that place, you could see oil, you know, settling. So I was curious to say, let's look at the microbial diversity of this environment over the years after a lot of spills. Sometime in 2011, I presented a monitoring report from this place, microbial, during the fourth European Bioremediation Conference in Greece. And I was surprised that some people from Austria, after my presentation, also presented a paper that 100 years after a certain spill in Austria, that there were still cases, that there was still a report of that place not cleaned up, 100 years after a spill. And so while I was scared, afraid that 10 years after a spill, we still had residual oil in that environment, the place did not recover, I was so relieved to hear some other people present that after 100 years in Austria, there was still, so that made me curious. I now needed to say, beyond the culture-dependent techniques in determining the microbial diversity of that environment, let me use the 16S metagenomic study and tools to be able to see if I'll be able to look at the holistic biodiversity of that environment. And that pushed us to looking at where we could do the 16S genomic studies. And part of our visit, we've been in the US since the 1st of June, and a greater part of the period between June 1st and now. And yesterday, we were at Argonne National Laboratory here at Lemont in Chicago, trying to run through quite a number of samples from these sites. And we are collaborating with Argonne National Laboratory here in the US so that they will be able to do, we're looking at quite some, but we want to present the preliminary results that we obtained from the 16, both a comparative study of the culture-dependent techniques and the 16S metagenomic studies from these sites. So, the research objective, therefore, this study was aimed at identifying the microbial communities that inhabit the sediments of a Lentic ecosystem polluted by aviation fuel about 14 years ago, using conventional culture-dependent method, and then the 16S metagenomic study. Thank you. When we looked at all of this, when I wrote to Argonne, I had to mention to them that this is the very first time, to the best of my knowledge, that any environmental media of this will be done from the Niger Delta environment. And so they now invited us to bring the samples, and we've been working with them since the 1st of June. Ibeno local government, strictly on the coastal environment, lying between this latitude. Like I said, we have 31 local government areas and in Akwa Ibom State, but Ibeno is one of the 9 local governments within the Niger Delta environment that is producing oil and is hosting Mobil Producing Nigeria, a subsidiary of ExxonMobil, and some other oil companies, oil facilities offshore that place, apart from the Akwa Ibom terminal. That's the Ibeno local government. This is the Niger Delta, and the southernmost part of Nigeria. At different times, we've sampled the location. There's one sample time, there's another sample time, and we'll be sampling regularly to be able to look at it. So, did the serial dilution for the culture-dependent techniques, culture inoculated and incubation, maintain pure culture to be able to isolate and characterize the organisms using standard methodology. And then we also had to do the 16S metagenomic, using the 16S metagenomic tools following standard protocols of community metagenomic DNA extraction using the Zymo soil DNA extraction kit. We had to look at the purity and the success of our, back home there, we used electrophoretic system to be able to look at, so that we could, if we see visible bands, then we now know that the extracted DNA has been quite successful and is within the concentration that can be sequenced. But right here, within the period we came, because of the fact that we had to run large number of samples at the Argon lab, we didn't use electrophoresis, we had to use the picogreen reagent to test for the success of the DNA extraction. DNA amplification and sequencing was done using standard methodology. And like I said, the overall bioinformatics analysis was done using the standard packages from the BLAST and then the biogenomics package. Results of the culture-dependent technique. We had to use several nutrient media because we are looking at the holistic. We want to find out what kind of organism can we isolate from this place. So we were not restricted. We used as many as we could. These were the ones that we could list, but we used almost all the media that could be used in isolating microorganisms. Okay? But you could see that using the dilation factor, and usually when we are doing this culture technique, we normally say we count from plates that are between 25 and 250 per plate. Okay? Or between 30 and 300. So but from here you could see 1, 2, 3 were the only ones that we could say were within a countable range. Okay? Now we went on. From those 3, we could come up with, this is the microbial count, so to speak. And we had to go to characterize it using a standard Begis manual. And so these were 1, 2, 3, 4, 5, 6, 7 species of organisms that we could isolate using culture-dependent technique. But what did we have as a result? So we could, all of those 6, we could say 6 species, 5 genera, each of them from different family, and all of that. This from the culture-dependent technique. But let's look at the 16 metagenomic studies, what we did. The total reads of 20,157 reads. And then we had the reads that passed a quality filtering was about 93.2%. And so we went ahead to look at the various, classify the reads, under various taxonomic levels. And we saw the kingdom, these were the percentage reads that passed quality filtering. And then from the kingdom, because we didn't use ITS, we would have wanted to use them to also identify the eukarya from that environment, but we couldn't, because by the time we sent the sample out, they couldn't have the standard, the ITS standard. So all they needed to do was to run for prokaryotic diversity of that environment, which was the bacteria and the archaea group of organisms only. And so they could 86.5% of the total reads were bacteria. Archaea, we had 3.54, and then 9.95 was all classified, which I would presuppose this would have been the eukarya group of organisms, okay? And we went ahead to now look at the phyla classification. We discovered the top 5 of the 27. We had 27 phyla. The 16S metagenomics tool classified the organism from that environment into 27 phyla. And the top 5 were these: the Firmicutes, chloroflexi, acinobacteria, and then these others. These others is made up of all other phyla that are present in that environment, but they are less than 3.5%. All of that grouped together made up the other, which was a 14.5%. Okay? So we went ahead to look at the classes of bacteria that we found in that place. And we discovered that there were 57 classes of prokaryotic organisms present in that environment. And we looked at the top 7 classes. And they were listed here. Again, unclassified was 17.52% of the total rates. And so, we still went on because we had to run down until we got to the species level. We saw 109 orders of microorganisms in that environment. For the very first time, I was amazed that at a remote aviation contaminated site, 14 years after, we could have this diverse, order of microorganisms in an environment. And yet, the place is still as fresh as if the spill occurred yesterday. These were the orders of the prokaryotic organisms that we found in that environment, with 20.85% as unclassified. We went ahead to look at the families. The tool that we use identified 224 classes, 224 families. And the top 7 of the top 7 were this, and the number of reads, and the percentage of the total reads. We went further to look at the genera. 498 genera, and the top 7 were here. But let's get to the most shocking aspect of this revelation, this study. 718 species of prokaryotic organisms were found in this place that was remotely polluted 14 years before now. 718. That's not all. The shocking aspect of it is that none of these species were identified using the cultural method. Among the 6 species of organisms isolated using cultural technique, they were not among the top 7. In which case, when we are talking about biodiversity in a place, you discover that your biodiversity, what we identify as biodiversity of that environment depends, in microbiology, depends on what you could isolate, what you isolate. What you don't isolate, for you, does not exist in that place, using culture-dependent technique. That's why, when in 1985, the issue of the Great Plate Count Anomaly principle came up, that what you have in the environment is not all that you can get from there. It was a relieving thing, because some of us got into microbiology because we were told that microorganisms cannot be seen with the naked eye. And so what you cannot see with the naked eye, the only thing you could use is seeing it will be a microscope. But sometimes we've been told by that principle that there are some organisms that are viable but not culturable. There are some organisms that are culturable, but you cannot, because of environmental conditions, isolate it in the laboratory condition, under laboratory conditions. And we now discover that culture-dependent techniques can only reveal less than or equals to 1% of the biodiversity, of the total microbial diversity in an environment. And 1% is grossly unrealistic, grossly not representative. And so, I had to tabulate, isolate from the culture-dependent technique, the top 7 from the 16S metagenomic study. And you could see none here is repeated here. And so each time we're sitting down to look at it, we are sitting down in our laboratory and we're doing culture-dependent techniques. We are just doing an exercise. But the truth about it is that I want to recommend henceforth the use of sequencing, metagenomic studies, which will reveal, which will give to you, much more detail of the microbial biodiversity of that environment. That's why I'm excited to collaborate with Argonne National Laboratory, Jack Gilbert's laboratory, so that we could do. I'm believing that in the next few months, we should be able to have all the environmental media from the Niger Delta environment fully analyzed using metagenomic tools. As at yesterday, we had to extract 18 samples from various environmental media, and the results, I'm still awaiting the results in the next 1 or 2 weeks when the sequencing results would have been ready. And so, comparing culture-dependent techniques and the 16S metagenomic tools, you see the great difference. While this reveal one kingdom, the archaea and the bacteria, this was only the bacteria. Phyla, or phylum, 27, 2. Species, up to the species, 718 species, only 6. The difference is obvious and is clear. And so, in trying to find out the realistic ecological, status of an environment, whether polluted environment or a virgin environment, the tool to use is a metagenomic tools instead of a culture-dependent technique. In conclusion, only 6 species of bacteria, no archaeal representative were detected from the conventional culture-dependent techniques. 16S metagenomic analysis detected a total of 718 species, comprising both archaea and the bacterial and archaea diversity. None of the 6 isolates from the culture-dependent techniques were among the top 7 most prominent of the 718 species community members of the aviation fuel contaminated Lentic site. Comparing the efficiency of both approaches, therefore, this study confirms and thus supports the Great Plains Cat anomaly, as well as support the adoption of the 16S metagenomic and other complementary omics tools in investigating and determining the true ecological integrity of any investigated environment. We want to appreciate Dr. Francisca Nwakorie of, some people are shaking their heads there. Francisca Nwakorie of NIMA, Nigerian Institute of Medical Research in Yaba, in Lagos, who helped in doing the extraction and then sending the samples out for metagenomic studies. We want to also appreciate the Nigerian Institute of Medical Research, Yaba, Lagos, for releasing her to be part of this team. And finally, we have a team that is quite passionate about the pollution, polluted sites, and we are passionate about it. The Environmental Biotechnology and Industrial Pollution Control Research Group of the University of Uyo, who are part of this research project. Again, we are grateful to Argonne National Laboratory for being part of this project, and they are willing and ready to help us in seeing us through. Thank you very much.
Thank you very much, Professor Udo. We really appreciate it. We've come to the end of this section.
We do have any questions?
Yes, we're going to take some very few questions because we have only 2 minutes.
Yeah.
So we'll just take the questions, then he will now answer. So all the questions, we're going to take them.
Okay.
So.
Okay, we'll now answer. Okay, whatever way. Thank you very much, Prof. I noticed that as sensitive as this other method, the genomic method is, it couldn't detect the species you detected by the conventional method. Am I right? From what you listed there.
No, it did, but they were not. What we identified from the culture-dependent methods were not the top 7.
Yes, but they were there because what you listed there.
Let him notice so that he call the questions and then he will answer.
Okay.
Okay. Okay. So another thing, in connection with that, I was going to advocate maybe for any analysis we have to do, we could use both conventional and the genomics. We could use both, a combination of both. I think I will advocate that. Okay.
Yes, yes.
The second question.
Yes, I've got
Oh, thank you, Prof. Though I'm not a microbiologist, but at least it drew my interest. But I want to suggest something. There is, looking at what you've taught us so far, I think the topic here is not really appropriate for this presentation, because you seem to advocate the importance of megagenomics over the conventional method. I'm of the view that the title should be reviewed to reflect this. I don't know if you understand me. Because from the topic you have here, though it has, a part of it is on it, but you are more on the side of you emphasize more on the relevance or the importance of the megagenomics over the cultural method.
Okay,
Thank you.
Thank you. Any other question?
We'll make another question, and then we're going to do panel, okay?
Okay.
Yeah, my prof, I have to ask this. I'm a microbiologist. From the list of nutrient agar you mentioned, you said you use series of agar, but I think you are missing something. I expected you to use YEMC agar, that's yeast extract monitor, that can also help you to detect the presence of agrobacterium, Acinetobacter, and stuff, and some rhizobium also present there. Then from the blast analysis and sequence stuff, I also expected you to have given us a dendrogram, you know, showing the roots of relatedness of all these isolates you mentioned, and how the different clades and leaves specify their family, their order, their class, you know, and so on. And you go a long way to, even without any person going through all these, your data and the charts and stuff. Once the person gets a look at either the cladogram or the dendrogram, the person will be able to say, this is it.
Okay, thank you. Super.
This is not question actually. So from the pictures that you have shown, you know, what I understood is that in Nigeria, is in Nigeria, right? Nigeria. So in Nigeria, plants don't need water, only oil it seems, for their growth. Because they look very healthy with oil.
All right. Okay. Thank you very much. The first question was, as sensitive as the 16S metagenomic tool is, why wouldn't that methodology also identify the 6 organisms that were isolated during through cultural method? Like I said, like I tried mentioning, the 16S metagenomics identified 718 species. These 6 may have been among them. These 6 may have been among them, but they were not the top. They were not the top 7. What we were showing here is the top 7, the 7 most predominant. No, I said they were not among the top, the most predominant 7. Yes, they may have been among the 718 species. Okay. Oh, me. Okay. Now, the issue, yes, like you said, you're suggesting that both methods could be used. We cannot help but use the culture-dependent method in microbiology. Because when you are going to look at the targeted genome, when you are going to do a profile, a genomic profile of a particular organism, you just have to isolate that organism first and foremost. And there's no other 2 ways of getting that organism except to use the culture-dependent method. So we can't do without it. Both of them are there, and we will continue to use it. But the truth about it is that if you must get the holistic, a true ecological status of an environment, the culture-dependent method is not the best, because it will only reveal to you less than 1% of the microbial diversity of that environment. So the third question was the topic. The topic here is not You know, this is omics conference. And we want, yes, this omics conference and biodiversity conference, we want to show that at the particular location, at the polluted site, that the biodiversity, we have high biodiversity at the particular location, but our cultural technique, our cultural methodology could limit our vision of the diversity. That's the essence of this. So that's why we were talking about, that's why I had to use we had to use the topic, microbial diversity in a remote aviation fuel polluted sites. Just to be able to show, using 16S metagenomics, the diversity will be seen to be very high, but you could not culture it and get it. Question 4 says, the list of media would have needed to use the yeast extract monitor agar. Like I said, we didn't use the ITS because we never anticipated when we called One Korea. They said they won't be able to run the ITS, the eukarya. And so what we couldn't do, what we didn't want to do was have the yeast on the list here as being isolated, and yet using the 16S metagenomics, because we didn't use the ITS standard, we would not be able to show that. I don't know if you understand what I mean. No, Yes. Yes, but if we isolated yeast using the culture-dependent method, if we use that to isolate yeast from using the culture-dependent method, this other thing, without using the ITS standard, we wouldn't be able to get the equivalent of the eukarya.
Okay, guys, it's okay.
Okay. The question 5, the dendrogram, and then the other pie chart. Like I said, this is just a preliminary presentation. The results of all of this, like I told you, I just finished extracting on Friday. I finished extracting the last samples on Friday. These are only a preliminary result that we just presented. The rest of the results will be presented in subsequent meetings.
Nice.